Welcome to the University of the Free State.
Wa amohela ho University of the Free State.
Welkom by die Universiteit van die Vrystaat.
Siyakwamukela eNyuvesi yase-Free State.
Situated in the heart of South Africa, my university is a creation of the past, the present, and the future. Across our 3 campuses, 7 faculties have been home to some of the most magnificent minds. Everything we do is rooted in our values of excellence, innovation and impact, accountability, care, social justice, and sustainability. We have been changing the world for more than a century. We continue to pave the way and inspire a fresh stream of knowledge and skill to follow in the footsteps of our scholars and alumni. We hunger for knowledge that will help us to be better and to do better. Our facilities encourage excellence, pulling us towards the light of new insight and ways of applying what we know. We lead by example, advocating sustainability in everything that we do. While we are making a current impact, we are also steering towards the future with the knowledge and skills we need to take the lead. The next years will be especially significant in our growth as our Vision 130 unfolds, which will peak in 2034 when the University of the Free State celebrates 130 years of existence. Vision 130 is our strategic intent to reposition the university for 2034. Join us on this journey. Together, we will go further.
This is normally where I say, please be seated. Or should I say, please be standing? No, I'm joking. Good afternoon. Good afternoon. Thank you. I was worried that Professor Martin's inaugural will become a wake. This is a moment of celebration. We need to be able to show happy faces and a degree of happiness in the ways in which we speak to each other. Dear friends, colleagues, distinguished guests, ladies and gentlemen, all those that are gathered here, I don't know if there are any students in here, postgrads. They should be. If not, there would be a real problem as a university. For those students that are among us, an inaugural lecture is a very, very special occasion. It's both an auspicious occasion for the university, but it's also an auspicious occasion for the individual and their family. It allows us to acknowledge the appointment or the promotion of one of our colleagues into the ranks of a full professor, and to introduce them not only to the academy but to the broader community within and beyond our university. I wish to start by acknowledging those that are in the audience and to see that this hall has so many of us in our presence. Obviously, the gentleman of honor, the acting dean of the Faculty of Health Sciences, Professor Collier Janse van Vuren, the head of the department, Professor Chris Viljoen, colleagues from the Faculty of Health Sciences, and those that are from our own university across the board, our students, our postgraduates, and our undergraduates, if there are any. Because it's a special occasion for all of us, and in particular for Professor Martin, it's important for us to acknowledge his family. I don't know if Dylan and Latoya are here. Where's Dylan? Ah, the dark glasses. Their mom, Marcella, I hope she is around. His Kenyan family will be visiting and joining us online, and that I think that is the beauty of technology these days. His parents, Jane and Cyrus Nyaga, siblings Mike, Lena, Kasan, and his Pretoria family, Auntie Jemina, Yannick, and Mateo. As we would normally say where some of us come from, Karibu sana. Ladies and gentlemen, allow me to say a few words about Professor Martin Nyaga, because it really is a privilege for me at a personal level. It's a privilege because I don't take for granted that we get the opportunity of introducing and saying a few words at an inaugural lecture of one of our colleagues. The inaugural lecture today, I look forward, among others that I have heard from him, Genomic Sentinels, charting the evolution of enteric and respiratory viral threats for Africa's health security. For those that are Africans at heart, this is an important lecture. Because Prof. Martin, you have now become a full professor of the university, I believe I can now call you Mzee. Mzee Martin, it is for us to come and celebrate your life's work in culminating with the title of full professor. It is about you this evening. And just to give you a sense, even though our acting dean is going to give us a full sense of who our colleague is, but just to give you a sense and why I'm so happy to be able to say a few words this evening. Our colleague, Prof. Martin Nyaga, is, some would say, an emerging. I would say no, he's no longer emerging. He has long emerged. He is a medical virologist of note. He is somebody who has earned his stripes over the many years. Having completed his medical virology and PhD in medical virology from the then Medunsa, the Medical University of Southern Africa. He came and joined us, and we are grateful that you've chosen us as your home. We know him as the head of the Next Generation Sequencing Unit within the Faculty of Health Sciences. And under his leadership, the unit has grown into a center of continental importance. And when I'm talking about a continent, I'm talking about our continent. And is now recognized as the WHO Collaborating Center for Vaccine Preventative Diseases, Surveillance, and Pathogen Genomics. We just came out of COVID, and it is because of the work that he and his colleagues did that helped us to deal with that particular pandemic as a continent. Looks like there are going to be a lot of claps tonight. Currently, he leads two major Gates Foundation-funded studies, one investigating the long-term effects of the Rotavirus vaccine, and the other focusing on sequencing the antigenic cartography of enteric viruses across 5 African countries: Cameroon, Ghana, Malawi, Kenya, and here at home. His scholarship has earned him many recognitions, wide recognitions. He is an NRF and NRF B-rated researcher. Just to give you in context, and Glenn can confirm, there are less than 10 B-rated researchers at the university at the moment. If I'm wrong, you can correct me. So he is in a very rare category of individuals. We have 2 A-rated, and so there is no question that, and no pressure either, that in time to come, our colleague will be A-rated. He is a Callistus Juma Science Leadership Fellow. I do not know whether it is 150 or 160 peer-reviewed publications and counting. I don't know how many thousands of genomic sequences which have been submitted to GeneBank, and 30 postgraduate students and counting. Professor Nyaga's story is a reminder that science, and I think this is for me what is important about why we do what we do. Science is the most powerful when it connects with humanity. We don't do what we do for the sake of just doing it. We do it because we love it, but we do it because it changes people's lives. So tonight we applaud Professor Martin Nyaga, and in good tradition, it is my pleasure and privilege to ask the Acting Dean to formally introduce our colleague to the audience.
Thank you very much. And indeed, ladies and gentlemen, a huge privilege for us to share this very special occasion with Prof. Martin, with his family, with his friends. And I think you've heard about everything he has done and the humble person he is. He has asked that I also acknowledge a couple of very special colleagues who has assisted him on the journey that he's going to share with us tonight. So before I introduce him, I just want to do a couple of further acknowledgements. Firstly, to his family that were already welcomed by Dr. Kubela, but then all key guests for him in person and virtually, including but not limited to Prof. Maringa, Attorney Sindiswa, Mr. Muruti, Mr. Sankale, then all his mentors, his co-investigators, specifically in the 2 collaborations that Dr. Q also spoke about, Prof. Duncan Steele, Prof. Mapalele, Prof. Mwenda, Prof. Jere, Dr. Dennis, and all other collaborators that's here present today. Then there were also a couple of institutional senior colleagues that have supported him, specifically in the growth of the UFS-NGS Unit since his inception in 2016. And that is also a very important date. If you listen to the lecture that he's going to share, the achievements that he have had in a very small span of time. Prof. Gert van Zyl, our Dean, who can unfortunately not be here tonight, and send his apologies and his best wishes, Prof. Martin. Prof. Corley Vatoon, Dr. Glenn Taylor, who is with us here tonight, Prof. Dani Vermeulen, and then also Prof. Chris Viljoen joining us here in front tonight. A couple of very special guests that have joined Prof. Martin on his journey leading up to this special occasion. There's a couple of highlights, and Dr. Kubela touched on a couple of them, but that I would also like to highlight. Prof. Martin has managed to be promoted from lecturer to senior lecturer, associate professor, and now full professor within 9 years. Colleagues, that is absolutely amazing. During this time, also becoming an NRFB rated researcher, as already mentioned, but then acquiring several international and national research awards and recognitions. The designation of the UFS-NGS Unit as a World Health Organization Collaborating Center for VBD Surveillance and Pathogen Genomics. Founding the AEVGI and SICV research groups, and being a co-PI on both those research collaborations. Being selected as a fellow of the prestigious Gates Foundation, supported Calestis Juma Science Leadership Fellowship. And then also closer to home, being an alumnus of the Transforming of the Professoriate Mentorship Program of the UFS. And I know that a special invitation was also sent out to the fellows, so there might also be other fellows from this program joining us this evening. So colleagues, if you use all of what has been said as a backdrop for the lecture, Prof. Martin, your lecture titled Genomic Sentinels, Charting the Evolution of Enteric and Respiratory Viral Threats for African Health Security, will deliver a comprehensive and an inspiring account of his pioneering work in pathogen genomics, its profound impact on public health in Africa. And it will share a visionary roadmap for the future. Another extremely important aspect of what he lived for is not just to change the world, but to also think about the future and prepare for the future. He will set the stage by acknowledging the significance of the occasion for him as a scholar, but also then immerse us as the audience in the persistent and evolving challenges of global viral threats, articulating the critical need for sophisticated tools to combat these pathogens, and establishing genomic science as a cornerstone of modern public health, taking us into the future. His profound commitment to mentorship and capacity building will emerge as a central theme, and I think it links so well to the question Dr. Kubela asked about students in the audience. And I think there are so many that will look up to you tonight and acknowledge you as their mentor later in their lives, as you are doing tonight. He will articulate his philosophy of investing in human capital, showcase his extensive supervision of postgraduate students and fellows. He will highlight how his efforts have directly contributed to equipping a new generation of African genomic scientists and building robust research infrastructure. And then definitely conclude with an inspirational message about empowering the next generation of genomic sentinels, asserting that a scientifically sovereign Africa is not merely a regional aspiration, but an indispensable pillar of global health security. Colleagues, and I think if we listen to this, we realize the change that Prof. Martin is making, not only for Africa, but in the world, and here amidst all of us, and we are so privileged. So family, friends, colleagues, please sit back and enjoy the absolutely amazing journey of excellence that Prof. Nyaga will now take us on, as he reflects on his career, his research, and his achievements. Prof. Nyaga.
Thank you so much. Thank you so much, Prof. Korea and Dr. Morabo, for that very generous introduction. Esteemed members of the Rectorate present, distinguished members of the Senate, deans of faculties, esteemed colleagues, students, cherished family members, including my two kids, Dylan and Latoya, and their friends Soira and Mateo is coming, and all my honored guests. It is with profound gratitude and immense humility that I stand before you today to deliver this inaugural lecture, making a significant milestone in my academic journey, and indeed for the collective pursuit of scientific excellence at the University of the Free State. In an era defined by rapid global health security issues, through persistent emergence of novel pathogens, the imperative to understand, track, and also mitigate viral threats has never been more urgent. Today, I invite you on a journey through the evolution of viral surveillance, culminating in the transformative journey or role of genomics, what we are calling the science of genomic sentinels, to solve and secure health for Africa and beyond. The outline of my lecture today, I will look on an overview of viral threats. Then I'll talk about a snapshot of my educational journey. Then discuss my core research areas, including the African Enteric Viral Genome Initiative, and also the sequencing of the antigenic cartography of enteric viruses. I'll further discuss the role of the unit as a designated WHO collaborating center for vaccine-preventable diseases and pathogen genomic surveillance, and also my passion for capacity building. And lastly, I'll talk about my vision for the future. Recent history, particularly the profound impact of COVID-19 pandemic, has underscored that infectious diseases know no borders. They are constantly evolving, and they are a challenge to global health security. Africa, we're very rich in biodiversity and also human capital in terms of the human potential, bears the brunt of many viral diseases, from endemic infections such as HIV, AIDS, TB, and malaria, and also from recurrent outbreaks such as Ebola, yellow fever, and Lassa fever. Traditionally, our diagnostic arsenals have relied more on targeted methods such as PCR for known pathogens and or serology for past exposures. These methods present a significant blind spot when confronted with novel viruses, highly diverse strains, and also complex co-infections. We must therefore move beyond reactive containment to proactive intelligence into anticipating and understanding viral threats before they become or escalate into widespread crisis. Now, I'll look at a snapshot of my academic journey, which actually began with a foundational curiosity in biological sciences, leading to me partaking a bachelor's degree in biomedical technology, and later on, I did my honors and masters in medical virology. But it was during my doctoral studies that I truly immersed myself into the rising field of genomics, recognizing its immense potential and appreciating viral pathogenesis. My experiences as a doctoral research fellow at the John Craig Venter Institute in Rockville in the US, and also my time as a visiting postdoctoral research fellow in KU Leuven in Belgium were very transformative. And it's during my time at Medunsa, or the SMU, which is a diarrheal pathogens research unit, that I was lucky to coordinate the African Rotavirus Genomic project that directly exposed me to applying advanced techniques to address critical public health challenges in our continent. When I graduated with my PhD in 2015, there had emerged a need for a dedicated state-of-the-art facility capable of performing next-generation sequencing here at the University of the Free State. And I had the privilege of leading the establishment of this UFS-NGS Unit in the year 2016, which it was and still is a significant institutional investment aimed at building indigenous capacity in genomic research. From its inception, I was very clear that I wanted the unit, among others, to sequence RNA viruses and also other pathogen genomics, including what we refer as targeted metagenomics. And this laid a groundwork for rapid response of viral threats. This initiative was not merely about just building capacity, but it was more about fostering expertise, developing robust workflows, and also creating a hub for genomic intelligence that could serve the university, the country, and the continent. And by last year, that is 2024, the UFS-NGS Unit actually automated all its liquid handling, and we acquired a very robust high-throughput sequencer, defined as the NS6 2000, which now we are using for most of our genomics work. Now I want to discuss about some of my core research, and I will start with the African Enteric Virus Genome Initiative, or simply referred as the AEVGI. This initiative has been very crucial in tracking rotavirus evolution in the pre- and the post-vaccine introduction for many countries, including Ghana, South Africa, Malawi, Kenya, and Cameroon. We all know that rotaviruses remain a leading cause of severe diarrhea and mortality in young children globally, with the most burden of disease being in countries from sub-Saharan Africa. And recognizing the need for continuous genomic surveillance in the post-vaccine introduction era at whole genome level, I founded the AEVGI together with other co-founders and co-principal investigators from these countries. In Malawi, we had Professor Kuswa Ujeri. In Ghana, we have Dr. Dennis Francis. In Cameroon, we have Dr. Farentine Gum. And later on, we included a co-principal investigator from Kenya, Dr. Julius Mugweru. The strategic goal of AEVGI was to investigate the long-term effects of the monovalent rotavirus vaccine, defined as Rotavirus. And the impact of AEVGI was so good. And through this initiative, we were able to track the emergence and the preference of rotavirus strains after vaccine introduction at all genome level, identifying the shifts in genotype constellation distribution. We reviewed a significant reassortment event between human and animal rotavirus strains, which is a key mechanism of viral evolution that can lead to novel genotypes with potential implications for vaccine efficacy. Further, we have been able to understand these evolution dynamics, and our findings directly inform strategies to optimize rotavirus vaccine efficacy and to ensure continued protection for vulnerable children in high mortality settings. This initiative has already yielded more than 40 publications in top-tier journals, contributing significantly to the global understanding of rotavirus epidemiology. We have also trained multiple postgraduates at master's and PhD level, and also postdoctoral research fellows, and we've been able to offer some of them full scholarships. But most importantly, we have provided data that is vital to improve future vaccine candidates, and also to provide online genotyping tools, and also we have provided policy briefs and letters. So what we did between me and my co-PIs, we decided first we'll do country-specific studies, then amalgamate the data into bigger data sets where we combine the data for all the countries that are participating. Thank you so much. Okay. So one of my former PhD students, Dr. Peter Mwangi, was attached to this AEVGI project, and he worked with both what we refer as the G1P8s and the G2P4s rotaviruses. And we looked at studies that involved a long span of time. That is 7 years before vaccination and 7 years after vaccination. That's a period of 14 years. And our studies were very, we were able to demarcate what was happening in the pre-vaccine era. For example, you can see in this sub-lineage, that's where most of the vaccine or pre-vaccine samples were lying, whereas the post-vaccine samples were lying in this other subcluster or sub-lineage, what we refer as 4A and 4B. So we found that there were some amino acid changes that were driving these differences. For example, there was a glycine in position 162 for the pre-vaccine samples and arginine at the same position for the post-vaccine strains. And we found that while amino acid substitutions could be a strategy to cope with population immunity following vaccination, selected amino acid residues were observed prior to the vaccine introduction, suggesting that there was natural evolution dynamics of wild-type RVA or rotaviruses that were likely to be involved. Further, we had a lot of reporting of globally of shifts of genotype constellations after vaccine introduction. So we wanted to determine whether this vaccine was due to vaccine pressure, due to vaccination or introduction of vaccination in these countries. In South Africa, we found that these atypical or not normal genome constellation were not necessarily driven by the vaccines because they were occurring even before the vaccination period. And in a similar study that was done in Malawi from our co-PI in Malawi, they found that there was a total shift of what was circulating immediately after vaccination. And for example, there were these G3 strains that were and disappeared for the last 20 years that re-emerged, and they not just re-emerged, but they came with many variants, 4 different variants, to be very specific. And now we looked at the aggregated data where we looked at all the samples together from the 3 countries. And this was done through a genome-wide evolution analysis of both the G1P8s and the G2P4s from the 3 countries, that is Ghana, Malawi, and South Africa. And G1P8s and G2P4s are globally important genotypes that fluctuate in prevalence. Vaccine introduction creates an immunological pressure on circulating viruses, and the aim of this study was to investigate if vaccine introduction created an immunological pressure that impacted the emergence and evolution of these G1P8s from the 3 countries. We sampled for a period of around 14 years, 7 years before and 7 years after vaccination from the 3 countries. It's important to note that South Africa introduced the rotavirus vaccine around the year 2009, whereas Ghana and Malawi introduced the rotavirus vaccine in the year 2012. So this shows how our correction was done, so that we could have a substantial amount of coverage of 7 years before and 7 years after vaccine introduction. So we went to the laboratory, we did our extraction, we did our cDNA synthesis, we created our genomic DNA libraries, and we sequenced using our very able platforms at the UFS-NGS Unit. For data analysis, we assembled our raw data using the Genius Prime. We created our phylogenetic trees using the IQ tree. We developed the temporal signals of each gene data set using Tempest. Then we estimated the rates of evolution, the times to the most common recent ancestor, and also reconstructed our relative genetic diversity using BEAST analysis. So what did we find? In Malawi, we found that there was an increase in diversity in the year 2015, around here, which was associated with a higher number of G1P8s that were detected and the emergence of a new variant. In Ghana, there was a decrease in diversity in the year 2014 to 2016, and this was due to very few sample sequenced or no data at all that was sequenced. In South Africa, the genes of these strains were under consistent evolutionary pressure, with gene data sets sharing contemporary times to the most recent common ancestor. And these strains appeared to have been introduced into the population approximately 2 years prior to the introduction of the vaccine. In Malawi, the strains exhibited similar evolution dynamics to those of South Africa, indicating similar evolutionary constraints that were occurring in different populations. And this was indicative of purifying selection, maintaining viral fitness. In Ghana, we found that there was differences between what was happening in Ghana from what was happening in South Africa and Malawi. And these strains from Ghana had distinct evolutionary dynamics, and then emerged more recently. Then we did selection analysis, and we found that there was selection pressure on variants within each country that varied, and biological implications were unknown for some of these same mutations. In conclusion, for these aggregated data from the 3 countries, we found that South Africa and Malawi share a similar genetic diversity of G1P8 strains with contemporary circulation of variants resulting in similar evolution dynamics. And we found that Ghana had distinct evolutionary patterns, and within country, there was selective pressure that varied. G1P8s detected decreased in the vaccine era from the year 2016 onwards, and this decrease was due to genetic diversity in strains in all the 3 countries. We do not see a sustained emergence of any new variants in the vaccine era, thus there was no evidence of widespread vaccine escape mutants. So building on the successes of the AEVGI, we went knocking again to Gates Foundation, and we were lucky to get another funding in September 2022 for a new project that we called the sequencing of antigenic cartography of enteric viruses. And this, or the strategic goal of this SASEF project, was to further optimize the efficacy of rotavirus vaccines by employing new approaches, including antigenic cartography. And these advanced techniques allowed us to virtually map the antigenic relationship between different virus strains, helping us to predict how well existing vaccines will protect against newly emerging variants. This work is very crucial for ensuring that our vaccine strategies remain effective in protecting the most vulnerable children in high mortality settings. Just like other studies, we did country-specific studies first before we conjoined the data. And I attached students into these studies. For example, for this work that was done on rotaviruses and also adenoviruses, astro and sapoviruses, Ms. Shange, who is my master's student, is reading this. And she, in this particular study that she worked on G8s, she found that multiple co-circulating G8 sublineages in South Africa were reported, and some of which have persistent for nearly a decade, suggesting that there was long-term endemic evolution. Further, the identification of significant amino acid changes in key antigenic sites were found, and these may affect immune responses to existing vaccines and could contribute to the virus's sustained circulation in the region, which highlights the role of interspecies transmission in driving viral diversity in Africa. Further, Ms. Shange has also looked at adenoviruses. I will give only these two examples. There's much more work that we are doing on this. Through a 13-year genomic study for these adenoviruses, which revealed these distinct evolutionary patterns between circulating human adenoviruses, serotypes, and we found that the human adenoviruses F40 strains remained genetically conserved, whereas the F41 showed greater diversity, including amino acid substitutions in key antigenic regions. These findings actually provide vital data for understanding adenovirus evolution in low and middle-income countries, with significant implications for future vaccine development and ongoing surveillance efforts. Similarly, my colleagues from Ghana and Malawi, they're also working on this. We have sequenced for them a lot of data on these 4 pathogens. And for example, this is a team headed by Professor Kusoyo Jerry through his 2 PhD students, Ernest and Frywell. And they have recently published genome announcements on noroviruses and also on adenoviruses. And there's quite a number of genome announcements that are coming before they work on the major publications. So my work is not just on whole genome. I've got a lot of interest on metagenomics to unveil the hidden virome, what is not known or normally diagnosed. So traditional diagnostics efforts and methods often only look for what we expect to find. But what about the unknown? What about the novel pathogens? What about the unexpected co-infections? My research on metagenomics of the gut and respiratory virome takes an unbiased approach to sequence all genetic material in a sample in order to discover the entire viral community present. This approach is very critical for resolving cases where traditional diagnostics fail to identify a causative agent, or for understanding complex interactions between multiple viruses. These projects are supported by grants from the South African Medical Research Council, from the Poliomyelitis Research Foundation, and also from the National Research Foundation. So starting with the enteric virome, enteric viruses are a diverse group of pathogens that may preliminarily infect the GIT. And globally, these viruses include viruses such as rotaviruses, noroviruses, adenoviruses, astro and sapoviruses. The impact is actually more felt in low and middle income countries, including some parts of Africa, and where acute diarrhea disease is the most recognized outcome. Emerging evidence suggests that repeated enteric infections can also contribute to long-term health consequences, including things like stunting and malnutrition. And understanding the epidemiology, diversity, and also evolution of these pervasive pathogens is critical for effective public health interventions. Our studies on enteric virome aim to establish the role played by the gut virome in young children over time, recognizing that early life exposures can have profound long-term health implications. And we have investigated how these viral communities may influence a child's health. And these findings open new avenues for understanding disease pathogenesis and also for potentially developing novel interventions to promote health child development. Just like other studies, I've attached students to this Enteric virome projects. And in this particular research niche that I'm very, that is particularly dear to me, on Enteric virome, the person spearheading it was my former PhD student, Dr. Milton Mogotsi, whereby he has published widely on these studies. But one of the key messages is that the gut virome of infants is dynamic and is very diverse, and this diversity occurs over time, with viral species becoming more prominent with age. And despite the absence of clinical symptoms such as diarrhea or vomiting, there is a significant high abundance of gastroenteritis-associated viruses over time. Moving on, I've got another research niche on metagenomics on respiratory virome. And we know that acute respiratory infections remain a leading cause of morbidity and mortality in children globally, and especially in low to middle-income countries. And despite their prevalence, the full spectrum of causative viral agents often remain elusive with conventional targets or targeted diagnostics. Our research leverages on shotgun metagenomics, which is an unbiased sequencing approach to comprehensively characterize the entire viral community, or what we refer as the virome, which is now present in these respiratory samples from children. And this methodology allows us to move beyond predefined targets. Applying this approach to studies here in the Free State, we have been able to identify a diverse array of respiratory viral pathogens in children under 5 years of age, including those without severe acute respiratory infections. So this respiratory niche is run by my former PhD student, Dr. Ems, and he has also published widely on this. But for example, in this study, we were able to provide remarkable pioneering data of RNA respiratory virome of children with and without severe acute respiratory infections during the winter and the summer of the COVID-19 pandemic. And these findings have raised concerns about whether some of these viruses detected with known pathogenicity may exacerbate clinical course or contribute to symptom manifestation. In this respiratory niche, we have also a study that was done by my former master student, Ms. Rengiwe, and we wanted to see whether there was an outbreak of respiratory syncytial virus during the pandemic. And we found that these strains that were causing the outbreak during the pandemic were genetically diverse, and they are forming small clusters, but they were not like new, variants, but because we could be able to match them with former strains that were detected before the pandemic. Moving on, I want also to discuss about our role in combating the COVID-19 pandemic. The sudden global COVID-19 pandemic presented a challenge and disrupted societies, economies, and healthcare systems worldwide. And the rapid spread of this virus underscored the critical need for immediate and precise identification of the virus, its transmission pathways, and also its evolutionary trajectory. Initial responses highlighted the limitations of traditional diagnostics and surveillance methods in keeping pace with rapidly evolving novel pathogens. And thus it became very clear that whole genome sequencing was not just a research tool, but an indispensable component of the robust public health responses. The scale of the COVID-19 pandemic necessitated a massively collaborative approach, bringing together scientists and institutions across South Africa. And as the head of the UFS-NGS Unit, I was a pioneer co-founder of the Network for Genomic Surveillance in South Africa, referred as NGS-SA in the year 2020. And the primary aim of NGS-SA was to rapidly generate SARS-CoV-2 genomic data in order to provide real-time information on how the virus was spreading, mutating, and also to map important targets for vaccine development. This initiative transforms South Africa's capacity for pandemic response, positioning South Africa at the forefront of global genomic surveillance efforts. The findings from the NGSA were published in substantially significant top-tier medical and scientific journals, including Science and Nature Medicine. And these publications involved extensive collaboration with global peers, underscoring the collaborative nature of modern pandemic response and genomic surveillance. These outputs fundamentally advanced scientific knowledge, providing crucial data on virus spread, mutation dynamics, and also targets for vaccine development, directly contributing to taming the pandemic. Among the most important discoveries that we made was the discovery of the Beta and the Omicron variants. And these variants, or these discoveries, provided crucial early warnings to the world, enabling rapid adjustments to public health measures, vaccine development, and also travel policies. Moving on, I want just to discuss about our role as a WHO collaborating center for vaccine preventable diseases, surveillance, and pathogen genomics. And as a WHO CC, we are tasked with 3 key objectives that directly impact public health across Africa. We are a trusted source of high-quality genomic data that contribute directly to the WHO global databases. And this data is essential for guiding public health policy and for developing new vaccines and treatment. This designation as a WHO CC for our unit opens the door for enhanced international partnerships. We work with a network of global experts to address health challenges like emerging infectious diseases and microbial resistance, and also other public health crises. We serve as a hub for capacity building, providing training and technical support to other institutions across the continent. And our goal is actually to empower scientists and laboratories to perform high-quality genomic surveillance, ensuring a unified and robust response to outbreaks. Similarly, we have had many projects through the WHO initiatives where I have attached several students to work on data from different WHO member countries where they have funded the data for us to work with at whole genome level. For example, Ms. Seposana, my former master's student, worked on data from Rwanda, which had introduced a slightly different vaccine, referred as Rotavirus, whereas also my former student, Ms. Makena Maringa, worked on data sets from Zambia, and Zambia had introduced the Rotavirus. So this work is actually attached to the WHO cohorts. But further, we collaborate with researchers or eminent researchers from this university, among them Prof. Trudy and our former postdoc, Ami, where they are looking, or we have sequenced data for them, not only on the human samples, but also animal samples. And they have actually determined that whole genome sequencing is a critical tool for identifying complex reassortment events and also characterizing the zoonotic potential of emerging rotavirus strains. These findings, done by Prof. Trudy and her team, underscore the imperative need for enhanced region-specific surveillance programs that create a One Health approach to monitor the evolution and the spread of rotaviruses across both human and animal populations. Further, we have extended our sequencing capacity to neighboring countries such as Mozambique and co-supervised their PhD students, in this case, Dr. Philomena, who we co-supervised with Prof. Inaugural. And these studies have gone to inform their public health sectors into making important decisions on how to introduce and also do surveillance for rotaviruses at whole genome level. So I would like also to touch on the impact of my research. The ultimate value of genomic surveillance lies on its ability to directly inform public health policy, moving beyond just mere data collection and actionable intelligence. As part of the NGSSA committee, I have contributed directly to technical ministerial report commissioned by the government of South Africa. Thus, I've been able to provide crucial advisory inputs on emerging COVID-19 variants from the year 2020 to the year 2022. Our expertise and our research extends just beyond SARS-CoV-2 and have contributed to a policy report commissioned by the government of Malawi on Vibrio cholera variants associated with the 2022-2023 cholera outbreak, where we demonstrated the transboundary impact of our genomic insights. These examples underscore the critical interface between cutting-edge science and responsive governance, where genomic data becomes a powerful tool for national and regional health security. In that respect, the African CDC appointed me to chair their vaccine preventable diseases focus group in order to develop for them a roadmap for genomic sequencing in the continent. I'm very passionate about capacity building, and this is how my current team looks. We have got a medical scientist and an admin officer, that is Milton and Mamero, and my current postdoc, Dr. Ems, the ogre in the unit, and my yeah, Dr. Ems is very instrumental member of the unit, and I hope we can be able to keep him for long. I'll talk to Gren on that. So the current PhD students, we have Linda, Young, and Ken. We have quite a number of master students. We have Tabisa, Somira, Angela, Torwana, Sesianda, Nkokozana, and Joyce. And our current interns, we have Surprise Mbali, Paresa, and Rengi. Rengi is actually more of a scientist than an intern. True. Then previously I've worked with a lot of people who have gone ahead, master's, PhD interns and also postdocs, who have gone ahead to take very good jobs. People like Prof Said, who is now a full professor. I passed through France and later on my unit. And now I'm very proud of this group that has helped us to go and become who we are today. My mentorship moves above just student supervision. And I try to hold big data and bioinformatics workshops, which have now become an annual event. And the potential direct impact of these workshops is to increase capacity for bioinformatics, to improve understanding of bioinformatics among researchers in South Africa, to increase collaboration between researchers in South Africa, and also to enhance international collaboration in the field of genomics. And the reduced impact being to increase demand for bioinformatics education, to increase development of bioinformatics, and also to increase the investment in genomics and bioinformatics research. Further, I am now approaching the younger minds at high school level, where from this year we started an outreach program to train high school runners. We had 9 schools visit our facilities in the university, and they spend a week running about genomics and getting, to learn about future careers in science, especially on the STEM units. So what is my vision for the future? Our future work will continue to expand the scope of metagenomic surveillance, aiming for unbiased discovery of novel pathogens and a comprehensive understanding of virome in diverse clinical, environmental, and even animal samples. We aim to move beyond single omics to integrate genomic data with other omics, such as transcriptomics and proteomics, in order to gain a more holistic understanding of host-pathogen interactions and disease pathogenesis. The sheer volume of genomic data necessitates that advanced computation approaches be applied. We will increasingly integrate AI and machine learning for faster analysis, predictive modeling of outbreaks, and also for more precise intervention strategies. Strengthening the pipeline from genomic discovery to actionable public health interventions to ensure that our research directly impacts patient care and community health. I can not forget to thank the people who have stood with us, especially our funders, the Gates Foundation, the African CDC, the World Health Organization, the National Research Foundation, the South African Medical Research Council, the Polymyelitis Research Foundation, and also the Diplomix SA. I could not have done this alone. I have people who mentor me, people I collaborate with, and people who I work with as co-PIs and consultants. I want really to mention my mentors, Prof. Duncan Steele, Prof. Jeffrey Mpadere, Prof. Jason Mwenda, Prof. Carl Kirkwood, my collaborators including Trudy, Prof. Sahid, Dr. Esona, Dr. Kwe, Prof. Pinch, Prof. Nikki, Prof. Mapaseka, Dr. France, Dr. Amand, Prof. Musyoki, and people who I work with almost on daily basis through our many initiatives, Prof. Kuzoa Jerry, Prof. Francis Dennis, Prof. Dr. Celeste Donato, Dr. Valentine, Prof. Kumwenda, Prof. Arox, and Dr. Julius. Further, I also want to thank my two pumpkins who make sure that I'm always awake. We make sure that I'm always awake to provide for them. Thank you so much, guys. Mateo, I recognize you. Mjomba, I recognize you. Thank you so much.
Colleagues, Prof. Martin has just also asked me to acknowledge his uncle and his cousin that had just arrived from Pretoria to be with us. So thank you very much. Martin, wow. That was awesome. Thank you. And I think, colleagues, you'll agree that it is very clear that in what Prof. Martin has presented this evening, he is a worthy full professor of the University of the Free State. While Martin was talking, it reminded me of a saying by John Nesbitt, who said, we are drowning in information, but starved for knowledge. Now, I need to give some context to this quote. This was made in a book he wrote in 1982 about megatrends, the 10 new directions transforming our lives. It was in 1982. It was a New York Times bestseller and on the top selling list for 2 years. But almost more than 20 years later, Clay Shirky offered a slightly different insight regarding this comment of we are drowning in information. And he said, it's not about information overload, it's about filter failure. And the reason why I raise these two quotes and share them with you this evening is because the work that Prof. Martin is doing is exactly addressing that divide. It's translational. It's going from just generating information to the next level of saying, how does this information translate? And how do we use this information in making decisions today about our healthcare systems tomorrow? One of the lessons that we learned from the COVID-19 pandemic is that we need to have accurate, timely information about the nature of the beast. COVID-19 wasn't the first pandemic, and it won't be the last. And in order to face the growing challenges in our healthcare system and future pandemics, we need to build the knowledge base, the capacity, not just to generate information, but also to filter that information and to be able to apply it in a way that allows us to support informed decision-making. If there's something that we learned about COVID-19, is that many bad decisions were made because of a lack of data. But we also have to be kind to ourselves and remember we did the best that we could at the time. Prof. Martin works at the cutting edge of viral genomics, which is a rapidly evolving field where accurate data and its effective use are increasingly vital for protecting human health. Beyond his contributions to COVID-19 research, his ongoing research on enteric viruses, particularly rotaviruses, as well as investigations into the gut and respiratory virome, are crucial for understanding their impact on child health and connections to metabolic disorders. When we think about Prof. Martin's achievements and research, Martin, when we look back in 20 years' time, which I hope we will have the opportunity to do, we will realize that the work you're doing now is the foundation for the type of research that we need going forward. And so we take immense pleasure and pride in Prof. Martin's achievements. Together with his team in the Next Generation Sequencing Unit, which has contributed immensely to, as he mentioned, the ministerial reports, to informing policy, to being designated as a WHO collaborating center for vaccine preventable disease surveillance and pathogen genomics. I think you will agree with me this evening that in his inaugural lecture, Prof. Martin highlighted the importance of robust data in being able to guide and support healthcare decisions, particularly when it comes to viral vaccine development, both for the present, but also for the future. And what stood out in Prof. Martin's inaugural lecture this evening is his commitment to expanding genomic surveillance, integrating multi-omics data, and harnessing artificial intelligence and machine learning for precision public healthcare. His commitment to capacity building and his commitment to leaving a legacy which will outlast his very successful career thus far is something that I think at the University of the Free State we can applaud. We are deeply proud of Professor Martin Nyaga. And Martin, no pressure, but we look forward to the next milestone of your career, and in terms of the work that you will do in taking not just the NGS unit, but viral genomics forward into the future. Thank you. I only have one more job to do, and that is to thank everyone for attending. Such an evening is such a highlight, and in Prof. Martin's life, I can tell you, it means a huge amount to have you present with him. And so I want to thank all the family and the friends and the colleagues who have taken the time to be here, and to everybody online who has also joined in through the power of the internet. And it remains for me to invite you to join Martin and his family for tea and refreshments in the foyer. But I would like to ask, because Prof. Martin is the man of the hour, that we remain seated and allow him a moment to take his place at the back of the hall, at the doors, so that you may also extend your congratulations to him when you pass through into the foyer. Colleagues, it's with great pleasure and great pride that I present Professor Martin Nyaga.